This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 403, recorded on August 19th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable yet simple to use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. Hi everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's probably about the same weather you have. It's sunny, some clouds, 83 Fahrenheit, 28 C, and actually not too bad.
It was. I just drove back from Ithaca, New York, and listen to this. We left at 7 a.m., dropped our daughter off for college. We left at 7 a.m. It was 59 degrees.
Wow.
And every time we drove through a little valley, it was full of fog. It was just amazing. And then we got down here. By the time we got down here, it was 82 and totally clear. Right now, it's 30 Celsius here and pretty nice out. Also joining us from North Central Florida, Rich Condit.
Hi, everybody. How you doing?
All right.
All right.
So what do we got here? We got 90 degrees Fahrenheit, and I'm just trying to figure out what that is. It must be like 32 Celsius. And dew point is 74. I don't know what that works out. That's probably about 50% humidity.
That's pretty. Like that. That's kind of muggy. It's probably like 60%.
Oh yeah, I mean, so our summer weather is hangs between dew point of 74 and 76. Ooh, yeah, it says humidity 56%. Mm-hmm. Okay, and it's summertime. That's what it is.
It is. It's summertime.
That's the way we do this.
So I mentioned that I just got back from Ithaca, dropped my daughter off at Ithaca College, where she's a freshman. Next week, our other son I'm bringing him up to Marist College, which is in Poughkeepsie.
Oh, yeah.
And then our oldest son is going to do his senior year. He's over at Stevens Tech in Hoboken. In like 2 weeks, no kids at home.
Wow.
Very strange.
That's different.
Very strange.
Yeah, very strange.
Do you remember those days, Rich?
It was gradual for me because... first of all, my son left 2 years before my daughters left. Mm-hmm. My daughters are twins, right? So they're the same age. But then our daughter, Sarah, went to school at the University of Florida.
Oh.
And so she was around for another 4 years. So it was gradual. My son left 2 years later. One of my daughters left 2 years later. The other daughter left. And so we, kind of had an opportunity to adjust. Plus, you know me, I'm so kind of wrapped up in my own junk and work and stuff that it was a lot tougher on my wife. I think she had to kind of remake her life. That's an exaggeration, but I think it was harder on her than it was on me. My biggest adjustment, to tell you the honest truth, is retirement. That's huge. Okay. Yeah. But we can probably do maybe a 2-3 hour podcast episode on that. Okay. We probably ought to stay away from that right now.
Yeah. If your kids are with you for 18 years and then they're gone, and
I'll tell you,
it's a little different. I got to admit.
So it comes full circle. As I told you guys when I was in New York, I'm moving to Austin, Texas.
Yeah, that's cool.
Okay, in the same neighborhood as one of my daughters.
Yep.
So I'm going back because that's it's like I'm really feeling the emptiness now with the daily job gone. So I'm going to remedy that.
Yeah, sure. I can understand that. Well, anyway, Alan, you got a number of years left, but
Yes.
It's weird. It definitely is strange. It was funny that earlier this summer I was telling my kids, go to college, get out of here. I've had it with you. But you realize, this whole time you spent together, well, it's different. It's okay. Life has to move on.
Yep.
So I'm at home today, so if I sound different, it's because it's a different room, a different mic, but hopefully it'll be clear and the virology will come through. And we have some follow-up for you. The first one is courtesy of Rich.
Yeah, so I wrote to John Carr, who was the PI on the bumblebee cucumber mosaic virus paper. I felt like, for me, I felt like we were kind of stumbling through pieces of that, though actually when I listened back to it, it sounds fine.
Maybe we were bumbling through it.
So that was mostly in my own head, but I wanted to make sure that he saw the podcast and give him an opportunity to comment if he wanted. Also, I was motivated by the fact that my middle name is Carr, C-A-R-R. So what the heck? So I wrote him. He wrote back, says, "Dear Rich, I enjoyed the podcast a lot, including the segment on the 2B paper, Plight of the Bumblebee. Love that title. You guys are the only commentators to spot the joke we slipped in about Oral-B. When deciding on what toothbrush to buy for the artificial experiments, Niels Groen, the first author, he hates his first name, Simon, and me simultaneously said we had to get the Oral-B. Niels also came up with the electrically actuated phrase as being suitably stuffy for a Cambridge-based paper. We also tried to slip in some 2B or 2B not to be puns, but could not figure out any phraseology that worked in the context of the paper. Assays tended to use 10 Bs. Apparently, car/cur is some kind of Old English Scots word for swamp dweller. However, my family came over from Ireland in the 1840s, something to do with potatoes, so that name might have a different derivation. Thank you for discussing the paper on the show. Best wishes, John." And that's John Carr with the Molecular Plant Virology Group at the University of Cambridge. So, yeah, actually, my heritage is somewhere back there in the British Isles. So that all makes sense. Okay.
All right.
Good.
So you wrote to him? Is that how
I wrote to him. Yeah, I wrote to him, and he wrote back.
Okay.
Alan? I'm wondering if we shouldn't skip to Steen's.
I just moved it up on the show notes.
Okay, good.
Because it makes sense. Yeah, Alan, can you take Steen's email?
Sure. Steen writes, "Dear corresponding author Vincent et al." That's good. "I think you succeeded in looking up the host range of cucumber mosaic virus quite quickly, TWiV 402, around 55 minutes. CMV is said to have the broadest host range of any plant virus. Question for you and your listeners. Is 1,200 known species the broadest host range for any virus?"
Wow, what a good question.
That is a good question.
It's got to be right up there.
Yes. I also think you did pretty well in the interpretation of the DCL1 and AGO1 mutant experiments. The difference between Cernas diced from stretches of double-stranded RNA and microRNA precisely processed from imperfect hairpins eludes many people, including some of the people annotating the relevant loci.
Can we pause there for a second? Because that's I'm not sure that's a distinction that we made well enough for the is there a word incognoscenti out there? That both of these involve small RNAs. One of them, the silencing RNAs, has to do with response to a virus infection, where a virus infects a cell, and the defense mechanism that involves silencing RNAs chops up the genome as a double-stranded intermediate, and then uses those RNAs as guides to go back to the virus and trash it. Whereas microRNAs are a cellular pathway. Doesn't have anything to do with a virus originally. A cellular pathway that we've discussed before that controls gene expression, but using small RNAs that hybridize to or bind to messenger RNAs. And some of the mechanisms involved in the two, there's overlap. Okay, but one is a cellular pathway, and the other is a cellular pathway, but specifically targeted to antiviral stuff. And so, since this virus protein can interact with small RNAs and potentially mess with both of those pathways, the important part of the paper was to try and distinguish which pathway was operative. Hope I did that okay. Anyway,
it made sense to me.
Okay, good.
Steen continues, personally, I'm not holding my breath about microRNAs directly triggering the volatile biosynthesis or emission. There is great potential for indirect effects with the pleiotropic mutant alleles the authors used. I'm not holding my nose either. The idea is plausible.
Pleiotropic meaning if you tweak one thing, there can be multiple, what would you call it, collateral effects.
Effects, right. Regarding tomatoes, not only can we find their wild desert ancestors, but we can cross them to elite varieties to potentially introgress traits like resistance to disease or drought. The tomato community, including my colleagues downstairs, benefit from some great mapping populations that have been developed. Finally, while cucumber was not the focus of the TWiV 402 paper, it might be worth mentioning that cucurbits, family Cucurbitaceae, interact with several significant viruses. A summer squash engineered for resistance to 3 viruses was one of the first transgenic crops approved in the U.S. in 1993. More recently, CRISPR-Cas9 mutagenesis has been used to induce point mutations in cucumbers that confer resistance to 3 related viruses, and gives a reference. The resulting amino acid substitutions in host translation factor EIF4E presumably prevent it from interacting with the viral genome-linked protein VPG, and thus disrupt translation of viral RNA. Similar natural polymorphisms confer virus resistance in other cucurbits, melon and watermelon, and this strategy could be used to recreate natural alleles in a variety of crops in a relatively fast and precise way. The cucumber varieties described in this work should be classified as non-transgenic. And Steen continues that would be non-transgenic in the U.S. anyway. I think German law currently would classify them differently because the authors did use transgenesis in the process of developing the varieties. Then P.S. I'd happily join a live chat room while you record TWiV if you started such a thing. I suspect, however, that live tweeting fact-check clarification requests would work better. Yes, I think if we were going to do live questions, Twitter would probably be the best medium for that.
So this bit about the transgenic, what was it?
Cucumbers or
with changes in the EIF4E is amazing. I think that's just so awesome. I looked at the paper. I'm thinking maybe it's at least a snippet, sure. if not a show. I mean, that's really cool. I'm also really interested in the fine-tuning of what legally is going to be described as GMO.
Right.
Right? If you go in and change a nucleotide using CRISPR-Cas9, I don't know, it's non-transgenic. Is there a distinction between non-transgenic and GMO? I don't know.
Well, yes. This is the fundamental problem of implementing an anti-science policy against a scientific progress, so you end up with absurdities.
Right.
So yes, virtually all of the crop plants that we eat are genetically modified in the sense that you wouldn't go out and eat wild tomatoes. You can't buy them for the supermarket. You eat the ones that have been deliberately inbred for things like disease resistances, and it's a question of whether that inbreeding was done in a Mendelian fashion non-precisely or done in a molecular fashion more precisely, and some people seem to have objections to one of those techniques.
I suppose the fundamental thing that people are going to a fundamental distinction is whether or not you introduce any foreign DNA into the plant. And so non-transgenic means you just went in and tweaked the existing DNA using, for example, CRISPR-Cas9, whereas transgenic means that you stuck in DNA from somewhere else.
That's right.
But then
it is. That's the original definition of transgenic.
I can go to the supermarket and buy a Tangelo.
Mm-hmm.
Yeah, right. So
which has DNA from a different species inserted in, and that was done through traditional plant breeding. I don't know.
Yeah. Well, the original definition was you use genetic engineering, the new field of molecular biology.
But what is that?
Where you take a plasmid and you introduce a gene on the plasmid into another organism. So that's why they call it transgene, although you can put more than
I think this is ultimately going to end up being like porn. You can't define it, but you know it when you see it.
Okay, I know what transgenic... We have our show title. I know what transgenic when I see it. By the way, if Alan sounds different, it's because he has a new mic.
I have a wonderful new microphone, yes.
You don't sound any different to me.
Courtesy of our sponsors and our listeners who've chosen to support us on Patreon. And this is the kind of thing that we can do. We can boost our audio quality.
We're going to boost everyone's audio quality, and eventually we'll do live streaming, a chat room while we record, so all our listeners or some of our listeners can interact with us, and we'll do all these cool things.
Right, because ordinarily when you do the episode from home, we have to use Google Hangouts, which means you can't do separate audio tracks. You get the whole thing in one commish, and the audio quality suffers. But this time we are recording our own audio tracks, and I'm able to do that because of this cool new mic. So thank you, everybody.
So
my computer says I'm recording my own audio. If you don't get a podcast this week, I failed.
Right.
Well,
I do have
a
Zoom
backup
saying it.
You got a backup.
Well,
good.
I'm recording everyone on one track.
Okay.
As well, so in case you always have to have a backup. Absolutely.
If the audio is not better, then it's Rich's and my fault.
Right.
All right, Kevin had written last time about chemists making videos. Kind of to accompany their papers, and he had left out the link to the YouTube video last time, and because we said, where's the video? I don't know, it's not here, and he writes and says, I forgot to include a link, and so he sends it. So thank you, Kevin.
And it's a good video, and it features the researcher is using biotin, and he uses this stuff from the supplement aisle at the supermarket, and it's labeled gluten-free.
Oh, what I like is he eats his reagent.
Yes, so he eats some of it to demonstrate that he just picked up this low-grade biotin at the supermarket.
I've never eaten any of my reagents, though I have to tell you, this wouldn't happen nowadays, but the first time I tasted squid was in high school biology. My teacher in high school biology, Gordon Chan, a wonderful guy, went out to the supermarket and bought a bunch of squid and brought them into class, and we dissected them all, and then he deep fried them. We ate them.
That's excellent.
Cool. All right, the next email is about something that, it's about the episode of TWiV a few weeks ago with David Tuller.
Which was outstanding.
Right.
I was not totally tuned into this, and it was just really, really well done. It was very informative and really important.
He's great. So David's been writing on my blog, as everyone knows. And this letter is about that podcast. It's from Gina, who writes, Dear TWiV at Microbe TV, thank you for continuing the despicable PACE trial flaws. So that's what David Tuller has been writing about, and Gina's a patient with CFIDs. She writes, Dave Tuller, you rock, as does this show, and talks a lot about her own illnesses. But at the end says, please, David, stay positive and passionate. Hope a Pulitzer Prize comes your way. Now, so David talked about the PACE trial for CFS, ME-CFS, which was done in the UK, I think in 2011, and which has been roundly criticized, that it was the subject of David's multiple blog posts at Virology blog. And now there has been a very important development. Now, we had actually not too long ago, David and I and a few other signatories had requested the data of the PACE trial. And this PACE trial, by the way, claims that you can improve patients with chronic fatigue ME by just having them exercise, because it's all in their heads, and all they need to do is exercise and they'll be fine. And the trial is full of flaws, which David has documented. We actually requested the PACE trial data from the relevant institution, which is Queen Mary University, London. They refused to disclose the data. So then the procedure in the UK, you go to the Information Commissioner, you file a complaint. Now, someone had done that before us.
Not to be confused with the Ministry of Information.
Right. So Alan Matthews, who had originally requested the data before us, we just decided we might as well do it and make multiple requests and so forth. He requested the data from Queen Mary University, London, of the PACE trial. They refused to disclose the data. Their argument, which they also used for our request, is that their release of the data would violate the confidentiality of the patients. So then Alan Matthews went to the Information Commissioner and appealed, and they last week released a tribunal released their decision, and they said basically Queen Mary has to release these data. There's no way it's going to compromise anybody's confidentiality. It's important that this gets out, and in fact, there's a decision which we'll link to from the first tier tribunal, which mentions the articles that David wrote on Virology blog as well, which is really cool. So pending additional appeals, which will probably happen, the data have to be released, and that's really good. And as David told me the other day, this could be the end of PACE because it's really not a good trial, and it needs to be either re-examined or retracted or in some way. Now, we also wrote, David and I and some other signatories also wrote a letter to Richard Horton, the editor of Lancet, and pointed out all these flaws with the trial. And he said, well, you know, these are good points. Why don't we publish your letter? So submit it as an official letter. So we did, right. And we waited 6 months, and they rejected it. And they didn't say why, but they had asked us to submit it, and then they said, no, we're not going to publish it. So this is an ongoing saga. Last week I wrote back to Horton and said, look, can you just tell us why you decided not to publish it? And in light of the recent tribunal decision, maybe you'd reconsider, because it looks like this PACE trial is this trial's coming down anyway. So anyway, I wanted to fill everyone in on that. I know not everyone who listens to the TWiV pays much attention to MECFS, but lots of people are very interested in this, and this is a big deal, this tribunal. So I hope that the data come out and people can look at it and say, this doesn't make any sense. We'll see.
So, what's up with Lancet? You know?
Lancet's weird.
Yeah. they've historically, they've certainly had their share of gaffes.
I mean, Wakefield.
Right,
yes.
Right.
Yeah, they're a medical journal, and they want to publish the hot papers, and sometimes when you want the hottest, most controversial paper, it's going to turn out to be wrong, and yeah.
What's most disturbing, what's as disturbing as anything else about this is that, damn it, when you get something wrong, when you make a mistake, you say, okay, I blew it, I was wrong, here's the right thing, okay? And nobody's doing that here. It drives me crazy.
Yeah, this trial is clearly flawed, and I don't know what Lancet is doing. Maybe they're trying to protect themselves from litigation, but I think this is going to come down, and they're going to someone's going to re-examine the data and say these conclusions are nonsense, or maybe not. We'll see, and they're going to have to do something about it. Because right now, physicians look at this and they say, okay, exercise, here's the exercise regimen, and it doesn't help people.
Well, certainly based on Tuller's analysis, I would say that as we go further into this, it's not going to get any better.
No.
Right.
All right. So
that's a good decision. Stay tuned to that, and David will certainly write about that. On to Zika. Here's our Zika sit rep. It's spread a bit in Miami, in Wynwood, and outside. Are there 30 cases now?
I can't remember where, but I saw some
I did
newspaper or something that was talking about 30 cases, yeah.
At some point, we're just going to, I guess we may stop doing the Zika sit rep when this just becomes one of those things that occasionally pops up in the U.S. and around the world. But for now, we certainly want to keep up with this, because
there's still things that happen which are really different and unusual.
Yes.
Right. So, the local transmission in Miami was a big deal.
Local transmission in Miami was a big deal. I'm waiting for reports of local transmission in other southern states, which are probably already occurring, but those reports just haven't gotten to the point that they're actually coming out.
Well, from my perspective, we have been saying since this started that given our understanding of the spread of this type of disease having cultural influences like air conditioning and window screens and et cetera, that it would, even though the mosquito range extends some distance north, that it would more or less stop at the border. But we don't know that for sure.
Well, and it's not going to stop completely at the border. It's going to be sporadic cases every wet season or every summer that are going to come around in the southern states, and it's going to be like dengue or chikungunya or West Nile, where this is something that happens.
That's our prediction, anyway.
That's our prediction.
My prediction, but until that's proven historically, 30 cases locally in Florida is newsworthy.
That is definitely new. I was not meaning to imply that this is something we should cancel now. This is certainly still trackable. Anyway, getting to it.
So another story. We talked last time about the genetically altered mosquitoes of Oxitec. These are males that are altered so that when they are released and mate with females, the offspring die. Last time we talked about the FDA approving trial releases of these mosquitoes, and in particular in the Florida Keys. But now we have an article from NPR, their blog called Shots, that says, because of the vocal opposition of people in the Keys, the local mosquito control board hasn't approved the trials, but they've put it on the November ballot as a non-binding referendum. So people are going to be able to vote on whether the mosquitoes can be released as a trial. Is that correct?
Yep.
Wow.
Yes, and a non-binding referendum, so the folks in charge, I guess, don't actually have to listen to it.
Right.
That's correct. That's correct.
What's the point?
Well, it gives people it's a formal way to collect public opinion on this.
It lets people vent, and I'm sure if there's a landslide against this, then the folks in charge, fearing for their reelection chances, would reevaluate. But I guess in the environment that we have now, this makes sense, because you might be out running or bicycling and accidentally inhale a mosquito and swallow it, and if it's genetically modified, then you just ate genetically modified food. So we all know how bad that would be.
So I wanted to remind people that we have a history in genetically modified mosquitoes and mosquito control. If you look back at TWiV 111, that's our live episode at Florida Gulf Coast University, where we actually interviewed people from the Florida Keys Mosquito Control District, and that's a nice description of what they're dealing with. Then in TWiV 375, about an hour and a half in, we talk about the science of the Oxitec mosquitoes. And I looked up the Oxitec page itself and give a link here to the science behind the Oxitec technology, and it's actually written up very well. You can start with that link and browse around the Oxitec site, but that describes the mosquitoes quite well.
This NPR article says, mosquito control in the Florida Keys is serious business, and the Florida Keys Mosquito Control District is one of the top control agencies in the world. So we had people from that district on that show, right?
Mm-hmm.
Yep.
Yeah, that's right.
And that was a great show, because I learned a whole lot about what they're doing, and it's an amazing operation. Very
good. All right. That's it for our Zika Sit Rep. Does anyone have anything else they would like to interject, or is that okay? You're okay?
I'm good.
All right, I want to tell you about our first sponsor of this episode, Curiosity Stream, the world's first ad-free nonfiction streaming service with over 1,500 titles and 600 hours of content, founded by John Hendricks of Discovery Communications. So what you're going to get is real science, not reality TV shows. You can get it on many platforms, including the web and all sorts of devices like Roku, Android, iOS, Chromecast, Amazon Fire, and Kindle, Apple TV. You can get it in 196 different countries. On the front page of CuriosityStream, when you first go to there, there's a little tab called Browse, and you have areas like science, technology, civilization, AI, communications, energy, engineering. And if you pick any of them, you get documentaries. Science, for example, is further broken down to biology, earth, genetics, medicine, mind, nature, physics, and space. So let's check out genetics here, see what we have. Okay, we have Enigma Man, a Stone Age mystery, viruses, destruction, and creation, the human face of big data, prehistoric autopsy, the science of walking with beasts. A lot of cool stuff here. You could really spend a lot of time looking at them, and they have other categories as well. So it's all.
So that viruses, creation, and destruction, is that a category with several things in it?
That's an individual movie. Some of them are categories. So there's one called Science Shorts. There are 20 episodes in there. Curious Minds, there are 2 episodes. But viruses, destruction, and creation. What is a virus? How does it affect humans? How does it spread? And they talk a lot about Zika. So this is real stuff. This is not fiction. It's the kind of stuff I think the listeners of TWiV and our other shows like. So you should check it out. Lots of good stuff. They also have interviews. They have nature, history, many more topics. There are monthly and annual plans available. They start at just $2.99 a month, less than a cup of coffee, or a single title on the competing on-demand platforms. So check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 months free of one of the largest 4K libraries around. Go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiM and the other Microbe TV shows. Now, over on TWiM, I had a complaint about running ads from someone. And I figure if there's one complaint, there are 100 people that don't like ads, or maybe 1,000. And I understand, but I'm experimenting with ads versus patronage, where listeners can decide to contribute. Now, we have a goal over on our Patreon page of $5,000 a month, which would enable us to do so many things, include hire someone to help in the production. And if we reach our goal, I won't have to do ads. But I have a feeling that the ads are going to be give us the most revenue to do what we want. And we do have a lot of wonderful patrons so far. But, we're patient. We'll wait and see what happens. But I tell you, if we reach $5,000 a month, I'll stop doing these ads. So you can have ad free.
Kind of like public broadcasting,
Exactly.
It helps to have some support for this. And every time one of the protocols that our public broadcasting does locally is they say, when we reach a certain goal, we'll stop this fund drive. Or if we can get people to sign up, and we won't have a fund drive. Okay.
Right.
So if you don't,
even NPR has ads.
Mm-hmm.
Right.
We thank so and so for their support, and that's an ad. So
yep.
So I don't want to be nasty here, but if you don't like the ads, go contribute to Patreon.
Yes.
Yeah, I want to be able to do more things. So when I get invited somewhere, I can ask the other co-hosts, "Hey, do you want to come and do this show?" We can travel and we can get equipment. We can get people to help us. So that's why we're doing it. And hopefully, if it becomes listener supported, we don't have to have ads. But for now, we use the ads, and they really help us do a lot. So, for example, that Harold Varmus episode was sponsored by PLOS Pathogens.
Right.
It allowed everyone to come together in New York City, so it's good stuff.
And realistically, we'll probably end up with a mix of the two, just like an NPR or public television.
I think so. Yeah.
We'll have some sponsors, and those ads will not be hopefully too intrusive, because we'll also have the Patreon support, so we'll be able to mix the two.
Yep. Now, I have a few papers here, which we'll kind of go over they won't take a long time. But they're all about vaccines. Alan had suggested a vaccine paper early in the week, and then I found that I had a few emails from people about other flu vaccines, and then I had a couple of papers in the folder, in the TWiV folder, about a vaccine. So I said, let's put them all together and do a vaccine episode. So that's what we got here. And the first paper is in the Annals of Internal Medicine, and very interesting. Sorry, it's the wrong paper. That comes later. The first paper is in Plant Biotechnology Journal, which you don't often talk about on TWiV. Cold chain and virus-free chloroplast-made booster vaccine to confer immunity against different poliovirus serotypes. Wow, what a combination of cool things there. Yes. So the background here is that, as everyone knows, we're almost at the point of eradicating poliovirus and poliomyelitis, and the use of oral polio vaccine, the live infectious attenuated vaccine, has really been instrumental. But now, as we get towards the end, WHO has recognized that we need to, at some point, stop using OPV and switch to an activated polio vaccine. How about that, Alan?
It seems like I've heard that idea somewhere before.
Alan and I, in the 90s, wrote a commentary in Science making this suggestion, and to which WHO said, never happen. You should never say never, I guess. So now we're doing it. I mean, part of the reason is that OPV can revert to neurovirulence, and you don't want to have that keep happening. It's easy to administer, but if you keep giving it to people, you're introducing viruses into the environment. So the WHO says, let's at some point stop using OPV, switch to IPV. However, IPV has issues as well. It's expensive. It has to be injected. You don't get gut immunity. It has to be kept frozen. So this paper, and I think it's a really cool find, Alan, tries to address these issues by producing a single poliovirus protein in plants.
So I want to follow up on the issues. First of all, I think one of the most important issues is that IPV does not confer gut immunity.
Right.
And which is important to make sure that there's no transmission of virus, and this addresses that as well. And the other thing is that IPV involves growing up gobs of wild-type virus, and that is a problem on an international scale. I have to say this paper, which, by the way, is open access, does a splendid job of summarizing all of those issues. It's one of the best summaries that I've ever read. It's very good.
Yeah.
Comes out of the University of Pennsylvania, CDC, and the Food and Drug Administration. The authors are Chan, Shao, Weldon, Oberste, Chumakov, and Danielle. And they basically have taken VP1, one of the capsid proteins of poliovirus. They have fused it to what is called a transmucosal carrier. It is cholera toxin beta subunit. It's a sequence that helps other things get across mucosal barriers. And then they produce this protein in tobacco or lettuce plants, but not just in the plant, but specifically in the chloroplast.
In the chloroplast, yes.
It's amazing.
This really interested me, so I looked into this a little bit. I think motivation for expressing in the chloroplast is that there are a number of genes there that are expressed at a very high level, and you can in fact get much higher level expression in chloroplasts than elsewhere. And apparently there are some other issues with expressing stuff in the nucleus that can interfere with expression. So there's the motivation there, I think, largely is quantity. In terms of the technique, they use biolistic chloroplast transformation. What that means is that they're using a gene gun, which is, for people who are not familiar with that, it is literally a gun.
Yes.
That shoots out these almost microscopic beads. I think they're gold or something like that.
Beads.
That are coated with DNA and actually mechanically forces these into the cells. The plasmids that are used, the transgenes, which in this case, as Vincent has already said, is a fusion protein between the cholera toxin B sequence and the antigen, the poliovirus capsid antigen, the plasmid contains that transgene flanked by chloroplast DNA to target it to the appropriate region of the chloroplast genome. The chloroplast, like mitochondria, has its own genome. So that's how it is that it's targeted to the chloroplast. There's another paper, if you look it up, that if you mix things targeted to the nucleus with things targeted to the chloroplast, those things will go to either the nucleus or the chloroplast, even in the same cell. So the targeting is accomplished by the flanking DNA. And I linked to a paper here that describes this and has a nice figure in it that shows this. And not only do they do that transformation, but then they have it. Part of it involves selection techniques and in vitro culture of the plants, and then further selections involving mostly antibiotic resistance, but also some culture techniques that manually select for things that are obviously expressing the transgene, for example, with GFP and that kind of stuff, to isolate plants that are homoplastic. That is, all of the mitochondria in the plant are expressing a transgene, not just a few of them.
Right.
Amazing technology.
It's very neat. So the VP1, polio capsid protein VP1, fused with this cholera toxin subunit, forms pentamers. So these are a structural subunit of the virus particle, forms pentamers, which should have the right antigenic structure to stimulate antibody response. And they show, as Rich said, you get lots of protein made. They get pentamers made. They bind the known receptor of the cholera toxin subunit, and they're pretty stable. They last a long time, up to 8 months at room temperature, I think. They don't fall apart. And the system that they test them in, the idea here is that we've been immunizing people globally with OPV for many, many years. A couple of years ago, it was found that if you give those same people a boost with IPV, they make lots of gut antibodies, even though IPV on its own doesn't induce gut antibodies against polio. If you boost a previously OPV immunized individual, you get a good boost in the gut. So that's one of the reasons why WHO has said, let's switch to IPV, because we'll be getting this gut boost anyway. Well, there are problems with IPV, as you've heard. So this preparation is hoping to replace that. So what they actually do, they use a mouse model to look for induction of antibodies. They actually they produce the protein in plants. They take the leaves, they freeze dry them and grind them up, and they either inject them or feed them to the mice. So they're basically feeding whole plant cells to these animals.
Right. They're giving them chewing tobacco.
Yeah.
You harvest it, you toast it, you dry it, and then you put a pinch between your cheek and gum, and you see if your mouse develops antibodies. And yeah.
And they do. So when they inject them, and what they do, they first prime them with IPV. They do subcutaneous, and then they go back with an injection of these plant cells, basically, and they get a really good titer of antibodies that will bind VP1. And if you don't give the IPV first, you get much less antibody production. And again, that is resistant to heating. That's stable at room temperature, that induction of antibody for a long time. Then they ask, if you give a mouse an injection of IPV and then feed them the plant cells making VP1, they make a lot of antibody, not only in the serum, but in the mucosa as well. They assay for antibodies in the feces. And again, if you don't give them IPV first, you don't get the gut antibody production. So it's sort of acting like the IPV boosting the OPV in the gut. This is boosting in the gut after IPV injection.
Right, and it's got. I mean, it's so close to being a really, really nice vaccine because you've got something that doesn't require a cold chain
Right.
You can grow it in tobacco and harvest these plants, dry them, store them. Obviously, the technology for drying tobacco is quite old, but you could do it in lettuce.
Right,
the point
right.
is that you do not have to refrigerate this stuff. You just stuff it in a container and ship it somewhere, and then you unpack the powder and rehydrate it and give it to somebody, and you can get a response. The catch is that they, at least in mice, you had to prime first with the injected IPV.
I think they ought to do this in marijuana. Okay.
Yes.
Because first of all, we'd have real medical marijuana then. Plus, it's going to take care of the anti-vax thing. Okay, because who's going to gripe about everybody's going to want their vaccines. A couple of other just technical things. They spend some time talking about codon optimization. They optimize the expression of this VP1 sequence for expression in chloroplasts, and that boosts their production 50-fold, so that when they're done, they've got like 4 to 5% total leaf protein. That is the antigen. I mean, that's incredible.
This is a great expression system, yes.
And they also talk about how advantages of the plants being that because of the cell walls, that assists in the oral uptake because the cell walls protect it through the stomach so the antigen actually gets to the gut. And my assumption is, I forget whether they state this specifically, is that part of the role of the cholera-toxin B protein is that assists in the uptake through the gut.
Right. Should also point out that when they look for neutralizing antibodies induced by this prime boost regimen, they neutralize all 3 serotypes. They're broadly neutralizing, which is terrific. Now,
that was a surprise to me. That
very surprising. Yeah, I don't know why that is. It's very surprising because
that idea is a new one on you, Vincent.
Well,
that cross protection.
Yes, I mean using VP1 from one serotype. They use Sabin1 VP1. I would not have expected it to have neutralized the other 2 serotypes.
Yeah, that was a little odd.
I don't know what's going on there. Now, there's several caveats. But the most important one, of course, this is in mice, and we don't know what's going to happen in people, as they admit. But they can do some phase 1s with this and just see if they are safe and they induce some antibody production. And you can look in the serum and in the feces in humans, and that would be very interesting to do. And we should point out, this isn't really new. This has been done before in plants, but this is a really neat application. And if this goes forward, there are lots of other vaccines that could benefit by a similar preparation like this. So this could really be opening the door.
And they did I mean, in looking at neutralizing antibody titers, they even comment rather sharply in their discussion on this. Unfortunately, most articles on plant-made vaccine literature report antibody titers, but not results on pathogen or toxin challenge.
Right.
So they are just highlighting the fact that yes, indeed, we did do the right experiment in this case. We looked to see that it neutralizes virus.
I tried to look that up to see if they did plaque assays, and they don't say specifically, but I assume they did. Because that would be the way to do that.
Now, I think to go into people, they're going to need some animal studies, right? So they're going to either need a mouse or a non-human primate of some sort, because you're not going to be able to go into people without that. But,
the irony of needing a clinical trial to go into people with tobacco.
Yes, indeed. But
we don't know if it's safe.
Yes.
To me, this is just incredibly exciting. I think we've done some of this before. I think we did a paper a while ago about making a universal flu vaccine in plants.
Yes.
But generally, I think that this plant technology for vaccines it has enormous potential. I'd really like to see this work out.
Well, and I think Dixon has mentioned, this is one of the applications of a vertical farm.
Mm-hmm.
Because you could grow these are the kinds of plants that you would certainly want to grow in a very controlled situation in order to get consistent results with your vaccine product.
Okay, now moving on to a few other vaccines. We got a number of emails about influenza vaccines. The first was from Chris, who wrote, Dear TWiVers, I'm a virologist with specialization in baculoviruses and ORF viruses. I have worked as a postdoc at the University of Otago under Professor Andrew Mercer for many years, but I've moved on. I'm now in Victoria, BC, Canada, and I stumbled across this news article from cbc.ca. I'd love to hear your thoughts. Please forgive me if you already talked about it. I've missed a few episodes, though I'm back and proud. Warmest regards to all the TWiVers. So he sends a link to an article from CBC News Canada. Flu shot effectiveness for 2015-16 disappointing data shows. So these are data presented by Danuta Skronowski to WHO some time ago, and it showed that in Canada the 2015-16 didn't show very good protection. So in 2014-15, the year before, the flu shot, this is the injected inactivated vaccine, showed essentially zero protection. And that's because the prevailing strain was different. But in 2015-16, the prevailing strain was H1N1. The match was good with the vaccine, but it was less than 50% protection, right? And that's not very good. So they've developed a way of looking at data that is really better than previous ways of measuring vaccine efficacy. Basically, they take a sample from every patient for lab testing. They also document whether the person gets flu or not, and then they can publish the data and look at it, and that's where they derive this conclusion from. Anyway, they don't know why it's poor. Okay, this is an issue with the vaccine. I don't have any data here, so we can't really look at it. But they did mention further down in the article the other thing revealed by the studies that
So the structure of this study, I'm not sure I entirely follow. They've got a population of patients, and it's this test negative design.
Mm-hmm.
Now they track influenza-like illness at doctors' offices, and they take a sample from everybody and test it,
mm-hmm.
and they record whether the person got the shot.
Mm-hmm.
So I'm not sure why that's a test negative design. I guess I'm confused by the name. Anyway,
right.
So they're looking for how often they showed up with influenza-like illness despite having gotten the shot.
Yeah.
Right.
Somehow this reveals variables that they weren't sensitive to previously, but without looking at the data, it's hard to know, and they haven't published it.
Well, correct me if I'm wrong, but historically, at least some time back, people just went mostly with company data that just reported seroconversion in people that were vaccinated.
But then there's also, and there's debate about what the right measure is. Mike Osterholm would say, unless you PCR amplified the virus out of the patient, then you don't know if they had flu.
Yeah, mm-hmm. Right.
So this falls prey to the possibility of a lot of influenza-like illness that is not influenza, I would have to see the paper to see exactly what's better about this analysis. Obviously, the Canadians seem to think that this is better, but I think it's important to point out that not all of the efficacy data from the past has been strictly industry sponsored. There have also been some independent efforts to track this, and they have come up with similar numbers to a lot of the industry sponsored stuff, but it's a very, very hard problem to track no matter how you do it.
Right, and an important thing is that there are different methods being applied, and so when you try and compare one study to another, the fact that the methods are different needs to be considered.
Yes, and that's a problem with a lot of the meta-analyses. You'll come out with a meta-analysis that says, oh, it's actually better than we thought, but then you have another one that says it's worse than you thought because the meta-analyses apply different standards to different trials. So it just, and as I think I've mentioned before, or we've talked about on TWiV before, what exactly do we mean by working for the flu vaccine?
Right.
So, you got influenza-like illness, and it turned out that you, in fact, were infected with flu virus, even though you got the vaccine. Would it have been worse if you hadn't? You know, if you hadn't gotten the vaccine, would you have been hospitalized or dead? And so we don't know to what degree the vaccine is working in addition to this. But if at least you do the study consistently, and I think the idea here is to try and maybe do this test negative design annually in Canada. At least I'm kind of getting that impression from this article. And if they keep up funding for that and do the same study year after year, you would at least get numbers that you could compare year over year, which would be useful.
So the Lancet just in August published a meta-analysis of these test negative design studies. Okay, I'm looking at the paper, and Osterholm is one of the authors on it. It's called Variable Influenza Vaccine Effectiveness by Subtype, a systematic review and meta-analysis of test negative design.
Okay.
And so the background is influenza vaccine effectiveness can vary by type and subtype. Over the past decade, the test negative design has emerged as a valid method for estimation of vaccine effectiveness. In this design, vaccine effectiveness is calculated as 100% times 1 minus the odds ratio for vaccine receipt in influenza cases versus test negative controls. I see. We did a systematic review of and meta-analysis to estimate, and their conclusions are influenza vaccines provided substantial protection against the pandemic H1N1, the pre-2009 H1N1, and type B, but reduced protection against H3N2. And their conclusion is we need to make better H3N2 vaccines. Now, that's not what was seen in this Canadian study. They said that for 2015-16 was not a good. It was a good match with the H1N1, but it didn't give good effectiveness. And I don't know the reason for that. I don't think they know it either. But what they do mention later in the article is that their data revealed that the live attenuated Flumist also didn't work very well,
right,
for several years. And that leads us to an email, two emails, one from Justin and one from Trudy. So Justin sent a link to a CDC news release back in June called the ACIP, the Advisory Committee on
Immunization Practices.
They voted down the use of live attenuated influenza virus for the 2016-2017 season. They say because data from previous years said it wasn't working at all. 3% vaccine effectiveness. So they said, we don't recommend that you use it anymore, and so that's what CDC published. And then Trudy sent us a link to an article. This study contradicts the CDC's advisory against using Flumist earlier this summer, and it might make for an interesting discussion. So this is a paper. I went looking for that. It's in the Annals of Internal Medicine. Live attenuated versus inactivated influenza vaccine in Hutterite children. All right, so this is a study where they took many Hutterite children, 1,186, 36 months to 15 years of age. They either got live attenuated Flumist or an injected inactivated vaccine, and they calculated the vaccine efficacy. Now, this is an interesting population because they are, a close-knit community members. They live in communities, are pretty isolated from everybody else. They often get influenza. So it's a good setting for estimating vaccine efficacy in a community setting. This kind of study is really hard to do anywhere else.
And the Hutterites, by the way, they're kind of superficially similar to the Amish in that they're a religious community that has remained isolated and relatively inbred for a long period of time, and has they live, I think, in Western Canada and parts of the Midwest. Whereas the Amish are more in Pennsylvania and Ohio, and they're different belief systems somewhat. I mean, they're both Christian groups, but this is a group that the important thing for this study is that they tend to keep to themselves.
So they got, as I said, over 1,186 children. They were randomly assigned to either getting, in a blinded fashion, either getting Flumist or inactivated vaccine. They got 70-some-odd% coverage in both groups. The influenza virus infection occurred at a rate of 5.3% in the Flumist group versus 5.2% in the influenza inactivated vaccine group. So in other words, they both protected. Now, there was no no vaccine control in this group.
Right.
And they say at the end, typically you get about 10% infection rates in unvaccinated people in these communities. So we don't know what the rate would have been in someone not vaccinated, but either way, they're both 5% with either vaccine. Right. So their conclusion is that LAIV isn't better than inactivated. But the other conclusion with respect to the discussion we've been having is it appears to work.
It appears to work, and it appears to be pretty much equivalent.
Yeah. Mm-hmm. So why this study is different from the ACIP and the CDC conclusion, we don't know. But, of course, populations are different. The viruses are different from year to year. It could be so many factors confounding this.
This study has, in terms of assessing effectiveness, the study has a different set of methods, a different structure, because they can look more directly at what's going on. Yeah. It's an actual randomized trial rather than just an observational trial. Okay,
and in past years, previously the live attenuated vaccine was specifically recommended for pediatric use because trials several years ago showed that it had this, incredible 80% efficacy in kids. So Flumist was a great idea, and then it suddenly stopped looking like a great idea, unless you're a Hutterite, which apparently, it works fine.
It's fine.
So the data are just kind of all over the place on this, and ACIP obviously has to make a very specific recommendation about a very broad population. And I think they were just looking at the past few years, and this is a vaccine like all the other major vaccines that shifted to a quadrivalent. Maybe that has something to do with it.
Yep.
And in any case, they looked at the most recent data, and the most recent broad population data suggests that the efficacy is abysmal. Now, it may turn out that it's better than that, or it may not, but ACIP, I guess, just had to make the call at the time. And
apparently they did 3 consecutive years of analysis, and they said, hey, we can't just go on for another 3 years. This is what we find.
Exactly. You have to quit that at some point. And now I guess it's up to the manufacturer to go and figure out what's going on. Yeah. And AstraZeneca is going to obviously they're going to be looking at this because this is an important product for them.
So there's a good article over at NPR Shots. The headline is "Study Says Flumist Vaccine Does Work, Contradicting CDC." And there's some good quotes from LJ Tan, who we had on Alan some time ago.
Yep.
Who basically says it's a tough thing to figure out, and there are different studies and so forth. But he says I could argue it both ways.
Yep.
And what do you do?
And I'm sure he has.
Yeah. So data outside the U.S. cannot be used to drive your decision here. Something is different between what we see in Canada and what we see in the U.S. We've got to figure it out, basically.
And ACIP, this is a group of people who they get together and they look at the data, and in many cases they're looking at exactly these same types of problems that we're talking about with all these studies and saying, well, we got to make a public health recommendation. Yeah. And so it's not necessary. Probably if you interviewed all the individual people on ACIP, they'd say, could have gone either way.
So a little administrative subtlety here. The ACIP, the Advisory Committee on Immunization Practices, is a committee within the CDC.
Yes.
However, this recommendation, it says CDC has recommended annual influenza vaccination for everybody. Oh, I'm sorry. Today's ACIP recommendation must be reviewed and approved by CDC's director before it becomes CDC policy, and they expect that review to come out in MMWR sometime in late summer, early fall. So ACIP has made a recommendation, but they're still mulling it over at CDC.
Right.
Yeah. So at the end of this NPR article, they say, in light of the Canadian data, ACIP may, consider it, but what are they going to do? Revise it before the season, or at the really start of the season, which is starting very soon now? And then, And then Tan says, if they don't use it another season, and CDC says the only data we want to look at are U.S. data, we're not going to have any data to look at because we're not using the vaccine. Because he said we're between a rock and a hard place.
That's why I say I'm sure AstraZeneca is pursuing ways to to look into this, and wherever they sell the vaccine, I would hope that they're going to track closely what the efficacy is, and if there's a problem, fix it.
Yep. So we don't have answers. We just have problems for you.
Well, I'm glad it's not my problem, boy.
Yes.
This is a tough one.
Yeah.
Well, which vaccine you going to get, Rich?
Well, I always get the injectable one.
Yeah, see, at Columbia, that's all they have left by the time I get it, so I get the injectable one.
I get the injectable because it's free at the town clinic.
Yeah.
My daughter has generally gotten the live attenuated because it's better, and obviously, she'd rather not get the shot. But last year, they ran out of Flumist, so she had to get the shot, and this year, nobody's going to be stocking Flumist, and it's not going to be reimbursed, so I'm sure she'll get the shot, too.
All right, our last vaccine story. Different virus, herpes zoster, and varicella zoster virus, I should say. So as you know, varicella zoster virus, if you get infected as a child, you get chickenpox, although we have a vaccine now, so you shouldn't be getting chickenpox anymore. But there's a large fraction of the population that had chickenpox as a kid, including me. And when you are infected, the virus goes latent in your dorsal root or cranial nerve ganglia, and then the viruses reactivate many years later, and you get shingles, the painful rash along your rib lines, which I've had about 10 years ago or 5 years ago or so.
Oh, how was that? I didn't know you had shingles.
I had shingles around TWiV number 7.
Oh.
I was doing a TWiV with Saul Silverstein, and I mentioned that, hey, I have shingles. It just popped up, It wasn't so bad. It's a little itchy, but there are far worse.
It can be awful.
No, some people get far worse complications, post-herpetic neuralgia, more painful problems. If you're immunosuppressed, you have even worse issues. So for this population who are at risk for shingles, a vaccine was developed a number of years ago. And it was developed. It's called Zostavax. It's a live attenuated vaccine developed by Merck. It was licensed for use in adults 50 years of age or older.
Now,
and it sounds like it's from Boston. Zostavax.
Zostavax.
You got to get your Zostavax.
Do I understand correctly that in fact is exactly the same attenuated virus as is in the chickenpox vaccine, but in a larger dose?
You got it. Exactly.
Okay.
That's exactly what it is, yes. It's a larger dose. It was tested in that age group, and it was licensed. Now, there's been some evidence that it's recommended you get one shot, 50 years or older, and initially the numbers look good in terms of effectiveness, but there was some indication that the effectiveness was waning over the years. So the first paper is a study in the Journal of Infectious Diseases, came out a few months ago. It's titled Declining Effectiveness of Herpes Zoster Vaccines in Adults 60 Years and Older. And this is an amazing study done at the CDC and Kaiser Permanente Southern California, which is a huge health organization, right? With hundreds of thousands of members. And what they did here was simply go through all the data and asked, they didn't have to do any experiments, any wet experiments. They pulled all the data on people who got this vaccine 50 years of age or over and whether they got zoster or not. And the numbers, 176,000 members were included in this study.
This is the kind of thing you can do with electronic medical records.
Unbelievable.
Yeah.
And what it shows is the effectiveness of the vaccine in the first year, 68%. And by the 8th year, it's 4.2%. And it declines slowly in the intervening years. So that's just with one immunization. So, if you get it at 60, 8 years, before you're 70, it's not very good at...
It's worn off.
At preventing zoster. And if you haven't had zoster yet, you're still at risk for having it.
Right.
So that's pretty eye-opening. And I don't know, actually, I suspect that many people don't get zoster in those first 10 years or so they're at risk for getting it.
So this makes me wonder, what sort of studies have been done, similar studies that might have been done about the effectiveness of the chickenpox vaccine given in childhood?
Yeah, I'm sure those are ongoing, right?
Because that's why I wanted to point out that it's the same virus.
So the question is, if you get that vaccine, are you not going to get shingles? Right?
Yes. Right.
So I've seen now studies to address that. We're not far enough out, I understand, and there are just some studies underway to look at that.
So wait a minute. Rephrase. If you get that vaccine, are you not going to get chickenpox?
That's another question, right?
Yes.
That's the one I want.
Right, but that vaccine is still new enough. I mean, it does seem to be, as far as I know, effective, at least in the kids who get it. We don't have kids getting chickenpox anymore who've gotten it, but the question is, when they get to be 20, 30 years old, are they still going to be immune? And then the other question is, when they get to be 60 years old, are they going to be immune from shingles?
Right.
I don't
know.
And
so
now wait a second. Shingles, though, is a reactivation.
Right.
Correct.
But the vaccine is an infectious vaccine, right? So it's possible that it establishes a latent infection, and you might get shingles. So
the vaccine could give you shingles, right?
Yeah. So, Rich, the 1 year, you get 2 doses of this chickenpox vaccine. It's between 85 and 98% effective at preventing chickenpox. Okay. But I don't know how many years they've gone out with that.
Does the vaccine strain establish latency?
I believe so.
I think it does, yeah.
Wow.
And so the question is it reactivate? And
yes, I believe the idea is that it being attenuated, if there were any reactivation, it wouldn't actually lead to shingles, at least a severe shingles disease. That's my understanding. But, we maybe need to get somebody on the show who can address some of these issues.
Yes.
Yeah, we'll get someone. It's very interesting.
The other thing that runs through my mind with this and all other vaccines is that in a given population, as you introduce the vaccine, you presumably reduce the amount of circulating wild-type virus. So initially, you may be vaccinating people, and they may be getting boosted because of circulating wild-type virus, but over time, that boost is going to go away. So things could change as time goes on.
Right. Okay. So
I've just found a paper from Journal of Infectious Diseases 2008 that looked at risk of zoster in adults immunized with varicella vaccine. They looked here. Right. You can't do a long enough follow-up on this yet, because vaccination only became routine in the U.S. in 95.
Mm-hmm. Yes. So we're not in our 60s yet.
So they looked at immunocompromised kids, and this is the long-term follow-up, and they find the incidence of zoster in that cohort is similar to the pre-vaccine era U.S. population.
Okay.
So it, I guess, can reactivate and cause zoster?
I think it's I think it's rare.
I think it's not common.
Yeah. So at some point, if most kids get immunized against chickenpox, we shouldn't have any more zoster, and we won't need a zoster vaccine anymore, right?
Right.
This is for the cohort of people who had chickenpox in the pre-vaccine era, and there's a lot of them around still.
Hopefully, we'll get mail.
Yes.
Well, I think we can get
somebody on
to talk about this. I've got a lot of people in mind, but what I wanted to talk about in connection was this: there's a phase 3 trial from GlaxoSmithKline. This is published in the New England Journal, and this is a brand new zoster vaccine that they're developing. It's a subunit vaccine. It consists of the varicella zoster virus glycoprotein E, which is an important. It's an essential glycoprotein, and it against which protective immunity is directed, plus an adjuvant called AS01. And they did a phase 3 trial in 18 different countries, 15,411 participants divided into vaccine or placebo groups, and then they follow them for a couple of years and see who gets zoster. Okay? Listen to these numbers. Overall vaccine efficacy against zoster, 97%. I
really like this. Yes,
that's
really
nice.
Stunned. This is a subunit vaccine with an adjuvant.
Right. So that means it's for the incognoscente here. That means we're talking about, instead of a live but crippled virus, we're talking about just a protein.
Right.
Just a protein. Now, the presence of adjuvant gives it side effects. There's pain at the injection site. A lot of people complained about that and so forth. And that's something that we don't use a lot of adjuvants here in the U.S. because of that, injection site reactions and so forth. But this is 2 doses injected in the muscle and 97% effectiveness. Now, we don't know the durability of that, which is an interesting question, but this is an ongoing study. But this looks great to me, and I've spoken with a few people who say this will probably get licensed at some point because this is a phase 3 study. It looks really good. It's better than the other one.
Well, now wait a minute, because the other one was, it's better in terms of the initial thing. The other one was 68% effective immediately.
Right. How are these people
waned
8 years later?
Exactly, because subunit vaccines, that's a problem with subunit vaccines, is that quite often the immunity wanes over time. So it's going to be important to look and see what the immunity is like in these people down the road.
But do you think we should wait 8 years to license this vaccine?
No.
It's okay to go ahead, right?
Yeah.
Yeah, it's a competing product, right? It fares slightly
absolutely
better, and if it's safe, seems to be, why not license it and see what happens? Yeah,
Yep.
And in 8 years, we do the follow-up study.
Right.
Anyway, this is a cool story, which I just wanted to point out to people, so there you go.
Yeah, I really like that.
I heard about this early this year, and I was stunned that this thing is almost 100% vaccine efficacy, and it's a glycoprotein. Those adjuvants, they really can boost your immune responses.
Mm-hmm. And this particular adjuvant, in fact, it's natural products, if you are into that kind of thing. It's very widely used. I don't think that there are really any issues with this adjuvant other than, in terms of long-term or serious problems. There are immediate some annoyance, okay? But I think given the benefit that you get out of it, it's okay.
Yeah.
All right, our other sponsor for this show is Drobo. Last time we talked to you about the making of apps to do different things with your Drobo. Remember, Drobo are storage arrays. You can connect them up to your Mac or your PC via USB 3, Thunderbolt 1 or 2. They also have network storage devices. We talked about Drobo apps that provide secure remote access. There's one of them called My Drobo. This is the foundation for secure remote access. It can do the following things. It can do DNS registration, register a unique URL endpoint. So you can select a username like vinny.mydrobo.com or alandove.mydrobo.com, and you can see it on the net. Of course, you can make it secure so that not everybody will get into alandove.mydrobo.com.
Right.
But it can be done. It does DNS management, keeps the DNS updated with the public IP. You can install an SSL certificate from a well-known service. It will install them and refresh them every few months, and so that ensures that the data are encrypted before it goes over the internet. And it also communicates with a relay server. The user's subdomain resolves to a server which forwards packets to the Drobo. So that's My Drobo, a cool way of getting your data. If Alan Dove is in Hawaii and he wants to get some data on his Drobo in western Massachusetts, he can do it. There's another thing called TNO. My Drobo is built using open source software to increase security and the probability of being bug-free. And the DNS and relay is a commercially hosted version of these open source programs. Drobo Access provides remote access for your files from any web browser. It's got a Dropbox-like interface. You use plain old URLs for sharing files. You can control links via passwords or setting a time for expiration. You get granular control over read-write privileges, version control to prevent overwriting a file, two-factor authentication. So you can do cool things with these Drobo apps. So not only is Drobo a cool storage device, but there are these apps that do really neat things. Twix listeners can save $100 off on their purchase of a Drobo Mini, Drobo 5D, Drobo 5N, or any 8 or 12 drive system. Just go to drobostore.com and use the discount code microbe100. You're still chugging along, right, Alan?
Oh, yeah. Yeah, mine is fine.
And what did you say last time? It's been boring for 6 years?
Delightfully dull experience. It's just what you want in a... Because my application for it, I'm not doing the sophisticated stuff with it at the moment. It's a backup, and it's a reliable... just a brick that sits on the back of the desk, and now the other one sits down in my basement, and they just back up my data, waiting to save my butt one day, which I'm sure they will do when I have a hard drive crash.
I'm going to try these apps so that I can access the data remotely, because I have mine in my lab, and it's got data on it, and if I'm home, I'd like to be able to access it. So
that could be pretty cool.
I'm going to try that, and I'm going to let you know how that happens. All right, we'll do one round of email, okay?
Sounds good.
First one from Paul to the TWiV. Listening to the Poetic Radiolab podcast, Cellmates, about the endosymbiotic theory of the origin of the mitochondrion, generated the following question. There are viruses that infect every living thing and some not living things. Where are the viruses that infect mitochondria? Where are the endogenous retroviral elements in mitochondrial DNA? A brief Google search turns up fascinating interactions between cellular virus products and mitochondrion function, but not viruses that infect the mitochondrion. Have I missed a memo somewhere? Thanks for the great work. There are no known viruses that infect mitochondria. They don't seem to be able to get there. They don't have endogenous retroviral sequences of any sort. However, if you look at the DNAs of mitochondria, of course, have their own genome. There are phage-like sequences in mitochondrial genomes. The implication is that they were infected many, many years ago when they were free-living bacteria. And many of these phage genes have gone on to become co-opted by the mitochondria. Like the polymerases, for example, come from phages. So I'm not aware of any viruses that infect mitochondria these days, but certainly they were when they were free-living.
And that kind of makes sense. You don't have viruses that target specific cellular organelles, because any virus that can get into the cell is probably going to be better served by targeting the whole cell. Yeah,
as far as we know, there's no mechanism for targeting, a mitochondria or even a chloroplast for that matter, right?
Right, and it's a little hard to see what the evolutionary advantage of that would be. I mean, other than...
Well, who are we to say, right?
Yeah, exactly. Next week, we'll probably be talking about a paper about a virus that infects the mitochondria that we've suddenly found out about. But yeah, it is cool, though, that there are phage-like sequences in there, which makes sense with what we think we know about the origin of the mitochondria.
Alan, can you take the next one?
Sure. Our friend Joni writes, colleagues, if I may. Yes, you absolutely may, Joni. Hank Green has produced wonderful science videos over the years. I particularly liked Crash Course episodes. This video is from a new production unit. In general, his presentations are interesting, entertaining, pithy, and well-researched. This episode presents some possible psychological constructs that may offer some insight into why some are afraid of vaccines. Best to all, with my hope eternal for spring 2016 in Cambridge, Massachusetts, and environs. I think we're a little behind on our email here. Currently 16C, cloudy with what appear to be stratocumulus clouds on a rarely seen blue background. But fear not. But fear not change. The forecast is only around 30% for sun over the next 10 days. Yeah, this was definitely a while ago. And she says, P.S. I subscribe to Curiosity Stream and pass the link on to friends. An extensive library, it appears.
By the way, folks, when we do ads, go click on them. They like that.
Yes, they do like that.
You don't necessarily have to subscribe, although they would like that. But if lots of people click from TWiV or TWIM or TWIP, they'll say, hey, those shows have great listeners. We want to support them some more. So click away. Like
now I assume I assume Johnny included a link with this email, which does not appear in the email anymore. But
I didn't have a link, no. But I guess you could search YouTube for
Hank Green. Hank Green has a bazillion videos on YouTube, which I was not familiar with him, but it does seem it looks like some pretty good stuff in there.
All right, I'm going to skip the next one because it's just about Jon Stewart insulting Donald Trump, and there's no science behind it. But the first sentence is good. He says, I want to say I'm going through the episode catalog once again, and maybe I will understand it eventually. You guys, sorry, Kathy, here in Indiana we use that word generically for a group of people. But Kathy would say, it doesn't bother me.
Doesn't bother her.
Doesn't bother her. All right. Rich, can you take Raihan's email?
Raihan writes, Hi, best friends for close to 8 years. I came across this cool phage and remembered your discussion of weird virus names many moons ago. Best regards, Raihan. And this is a link to Viral Zone to a family of phages called corndog-like virus.
Yeah.
I love this.
The phage is corndog. And actually, I've been familiar with this for a while. Roger Hendricks, I think, named this phage as part of his early sort of phage discovery program, and it does indeed look like a corn dog. It's got
it's great. It's great.
It's got this elliptical, I would call it sort of elliptical phage head. It's sort of an elongated thing, and a very, very, very long tail. So it looks like a hot dog on a stick.
It's got a delicious double-stranded DNA genome inside dipped in a batter of capsid proteins and then stuck on a tail tube and presumably deep fried in a mycobacterium. It's apparently there's a mycobacterium phage firecracker as well.
Wow.
That's
in this genus. Presumably
great.
With a similar.
Yeah, these guys. This is from the phage group in Pittsburgh. They've been having a good time with this for quite a number of years.
Clearly.
8 years, our friend. That's great, Raihan. Thank you. We appreciate it.
Thank you.
All right, let's do some picks. Alan, what do you have?
I have a video on Vimeo called To Scale. And it's a group of people building a model of the solar system to scale. So they start out with the sun is about the size of a small car. It's like a big inflatable ball. And they take that out into the Black Rock Desert in California because that's how much space they need. And they do this very, very nicely produced video with lights and time lapse at night showing them positioning the various planets and what size they are. And the Earth's about the size of a marble. And they fit this whole thing. And it's miles across at that scale, and they shoot video of it from out on a cliff overlooking the Black Rock Desert, so you can see this aerial view of it, and they drive the cars around to get light painted orbits of the planets. A lot of really cool stuff in there, and at one point, a guy stands, I think, at Neptune, and he looks back at the sun off in the distance, and that's how far away the sun would be if you were standing on Neptune. It's just a really neat illustration of the size of just our local piece of space.
Cool.
Having just been in Ithaca, did you know they have a Sagan planet walk there?
Oh.
It is a laid out model of the solar system at 1/5 billionth of the actual size. So they have the sun in downtown Ithaca, and you walk down the streets, and you go, through Mars, Earth, Venus, and Mercury are very close. You get the Jupiter, Saturn, Uranus, Neptune, and Pluto is at the edge of town. It's very cool. Yes, and I saw one of them the other day while I was walking. And, of course, all of that wasn't there when I was a student at Cornell. Didn't have anything. So it's the same idea, right? What's the scale on this one?
I don't know what the numerical scale is, but as I say, the sun is about the size of a small car, and the Earth's about the size of a marble.
Yeah, you have to. You have to compress it. Sagan Planet Walk. Cool. Rich, what do you have?
What I have, actually, I have a TWiV. I have a pick and a half. I have an informal one. I was going to pick, but I sort of picked it before. I have to say, as an update, that some time ago I picked the Expanse novels, science fiction novels, and the film that went along with that on the Sci-Fi Channel. At the time, I think I had only read the first of 5 novels, and I'm just about to finish the 5th one, and I can report that they're worth it all the way through. They've got me. Okay, so that's just a boost for my previous pick of The Expanse. For now, actually, I have an update of a pick that Dixon did a couple of years ago on TWiV 287. He picked Solar Roadways, and if you look back on that, in fact, that link doesn't work anymore, but if you look up, there's a bunch of links in my pick that'll work. So there's this invention, basically, that are these like pavers, if you like, for sidewalks and roadways that are solar collectors. So the idea is that you could pave a road or a sidewalk with this, and it would convert the they're like solar collectors and make energy from the roadway. And the idea is you can have solar panels all over the U.S. and generate like 3 times as much energy as we need to run the country just by paving the roads correctly. So this is a sort of an update on that, saying that in where is it? Is it? I'm sorry.
In Missouri, I think.
Missouri, on a just on one of the welcome centers in Missouri. They're going to go ahead and, as an experiment, pave some of the sidewalks in this. The fun part is it's Route 66. This is on Route 66. So the idea is that this historic highway through the U.S., Route 66, they're starting this experiment with paving with solar panels. They've actually crowdfunded this in part, and part they have a collaboration with the traffic people in Missouri. They crowdfunded $2 million to do this. So they're going to do the sidewalks around this welcome center, and they hope to do sections of Route 66 in the future. And in addition to collecting energy, these things are also smart. So they can be programmed to have lines or road signs or warnings or whatever. I just think that this is a wonderful
Or in a more
idea.
cynical view, advertisements.
Ooh, Alan, my goodness.
Sorry.
That is a little cynical, but there you go.
I was a little skeptical of this when he picked it, because I thought,
well, so
glass on a highway.
So was I, but this is a step forward. It's not actually on the sideway, on the highway. It's on the walkways. We'll see what happens.
Yeah.
Right. Well, there's you got to have ideas.
Yeah, and these people are promoting this at this welcome center are obviously passionate about it. This is great.
Got to have ideas.
It's a great idea,
and you got to have skeptics.
Yep.
All right. My pick is an article in the New Yorker. The Race for a Zika Vaccine by Siddhartha Mukherjee. So you can get the whole thing, the whole article, right online. You don't have to subscribe or anything. And Mukherjee, of course, the author of The Emperor of All Maladies, which is wonderful, and also a book about the history of DNA and the gene, which I picked some time ago. And this is just his typical style, which is so nice. He tells a little story, and he does it really well. So,
and if you read the online version, there's an animated graphic at the front that you'll either find gratifying or disturbing.
Yes. I can see for sure it going either ways. Anyway, well written. You can read it in 10 minutes or so. Check it out.
Yep.
And we have a couple of listener picks. Bo Don writes, this would be my pick of the week. Perhaps you've seen it. Penn and Teller do not mince words when it comes to vaccination. And this is Penn and Teller's pro-vaccination video, which has been picked a long time ago, but cannot be picked too many times.
Yes, this is a fun one.
And Bill writes, Dixon recently mentioned Burke's Connections TV show. The first series came out in the late 70s and was delightful. There were a few sequels. I still find the first series to be the best. If you haven't seen the first episode, The Trigger Effect of the first series in a few decades, you should go back and see it again. You will run into a couple of startling surprises you didn't catch during the Carter administration. Connections is available online. I suggest a double header of the first and last episodes of the series. And he gives links to the Trigger Effect, Yesterday, Tomorrow, and You. Those are the first and last episodes. And he also says, don't miss this amazing sequence. James Burke, classic, the famous rocket takeoff scene. Bill, science guy. Bill the science guy. All right, thank you, Bill.
Oh, he says science guy, backspace, folk.
Well, I don't get that.
So instead of backspace would be like he's backspacing over science guy.
Science folk.
Saying science folk.
Oh.
Science folk.
So caret
Yes.
H is backspace.
Right.
I didn't know that. Did you know that, Rich Condit?
No. I'm just really out of it.
No, you're not, but that's something that... Why would Alan know? Is that because you're a ham radio guy?
No, it's... I think backspace, I don't know if... current keyboards do the same thing. If you hit Control H, it gives you a backspace.
But he's a guy. It's okay. He's a science guy.
Yes, you can call yourself a guy.
If you're a guy.
If you're a guy.
If you're a gal, you could too if you want, right?
Yes, if you like.
All right, that's TWiV 403. We got 404 next week, folks.
Yes.
Dixon better be there for his little bit, the error code.
Dixon, yes. Next week is going to be in our normal place. We love getting your questions and comments. You can send them to twiv@microbe.tv. And if you go over to microbe.tv/contribute, we have several ways that you can support our shows. Patreon, PayPal, credit card, and so forth. Check that out. Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsors of this show, Curiosity Stream and Drobo. Please check them out. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.